Welcome to the Explainer. Today we are diving deep into a bacterium that is, I think, one of the biggest hidden threats in our field right now, Mammaliicoccus sciuri. Now, this whole explainer is based on a comprehensive Gemini deep search, and it's really tailored for my fellow medical microbiologists out there. And don't worry, all the sources are right there for you in the linked notebook document. So let me just kick this off with a question that really gets to the core of this whole thing. What if the original blueprint for MRSA, one of our greatest challenges, didn't actually start in Staphylococcus aureus? What if it came from a bug we've been dismissing for decades, a bug that's been hiding right under our noses? Well, the organism at the center of this whole story is Mammaliicoccus sciuri. And yeah, using that full name is really important. It represents a huge shift in how we understand this bacterium, and it's a story that involves a new identity, constant misidentification, and some frankly incredible genetic power. Okay, so first things first. To really get a handle on this bug, we have to talk about its new identity. This is way more than just a simple name change on paper. It's a fundamental clue about its evolution. You know, up until 2020, every single one of us knew this as Staphylococcus sciuri. But then a huge phylogenomic study came out and basically proved it was so genetically different from the main Staphylococcus group that it needed its own genus, Mammaliicoccus. The key takeaway here is that this isn't just academic housekeeping. This reclassification points to a deep genetic split that has very real consequences for how we find it and track it in the lab. So, this genetic uniqueness, this is where things get really tricky for us in the clinical lab. Because of its weirdly unique biochemical profile, this organism is an absolute master of disguise. Now check this out, because this is what makes it so fascinating. Most of the workup looks totally routine. It's a gram-positive coccus, it's catalase positive, it's coagulase negative. You know, it falls right into that big coagulase negative staphylococcus bucket. But then you hit the oxidase test, and it's positive. That is so unusual for anything in the Staphylococci family that it can completely derail our identification algorithms. It can make you think you've got a micrococcus or even make you second-guess your gram stain. That one test is probably the single biggest reason Mammaliicoccus sciuri is so massively underreported. So, okay, let's say we actually managed to nail the ID. What are we dealing with clinically? For years, this thing was just brushed off as a contaminant, but the case file is now overflowing with proof that it's a seriously dangerous opportunistic pathogen. And this slide just lays it all out. We are not talking about minor stuff here. Mammaliicoccus sciuri is a well-documented cause of bacteremia that spirals into sepsis, devastating infective endocarditis on prosthetic heart valves, and nasty peritonitis in dialysis patients. Make no mistake, when it gets the chance, it's a formidable pathogen. So how does it pull this off? Well, a huge part of its playbook is its incredible ability to form biofilms. This is the key to all those device-associated infections we just saw. It grabs onto a catheter, a prosthetic joint, whatever it can find, and builds this protective fortress around itself. Once that happens, it's almost impossible to get rid of with just antibiotics. And that's why source control, literally getting that infected device out of the patient, is absolutely non-negotiable for any chance of a cure. Okay, so we've established it's a pain to identify, and it's a biofilm-forming monster. But now we get to the most alarming part of this whole story. Its role isn't just as a pathogen itself, but as a gateway, a transfer station for antimicrobial resistance. This is the origin story that we almost never talk about. Pretty much every isolate of Mammaliicoccus sciuri out there naturally carries a primitive ancestral version of the mecA gene. But then it picks up the real, fully functional mecA gene on a mobile element. It effectively becomes a stealth reservoir, this genetic mixing vessel where these resistance cassettes can evolve and get stronger, and then it can transfer that fully loaded, high-powered resistance package over to Staphylococcus aureus, basically creating brand new lineages of MRSA. And as if being the ancestral home of mecA wasn't bad enough, this happened. Daptomycin. That's one of our last-ditch drugs for serious MRSA and VRE infections. We always thought resistance to it was this slow, painful process of chromosomal mutations. But the discovery of the DRC gene on a plasmid in Mammaliicoccus sciuri means that high-level daptomycin resistance can now just jump from one bacterium to another in a single step. I mean, this is a complete paradigm shift. It's a massive public health siren going off. All right, so when we're faced with an organism this challenging, this dangerous, how do we even begin to respond? How do we treat these infections? And here is the fundamental challenge for every single one of us, whether you're at the bench or on the clinical side. There are no species-specific EU cast breakpoints for Mammaliicoccus sciuri. We are literally forced to guess, to extrapolate, using breakpoints designed for other coagulase-negative Staphylococcus. This guideline gap means that getting accurate, comprehensive susceptibility testing from the lab isn't just a nice-to-have, it becomes the single most important piece of evidence to guide therapy. And when you look at actual treatment protocols, you can see this guesswork in action. The Dutch SWAB guidelines, for example, for really serious prosthetic infections with methicillin-resistant cons, they often recommend combination therapy like vancomycin plus rifampin. But then, international guidelines like from the IDSA might list vancomycin, linezolid, or daptomycin for things like bloodstream or skin infections. The point is, these are all based on general cons principles, not on hard data for Mammaliicoccus sciuri. Now, in kids, the core principles are the same, but the details become absolutely critical. Dosing has to be strictly weight-based. Therapeutic drug monitoring for drugs like vancomycin, it's not optional, it's essential to keep them safe. And we have to remember, this is a major cause of late-onset sepsis in the NICU. And of course, you've always got those age-specific rules in the back of your head, like staying away from tetracyclines in younger kids.
So, let's bring it all together. How do we prevent these infections from even starting? And what's the big picture takeaway from all of this?
Prevention, really, it all comes down to the absolute fundamentals of infection prevention and control, just executed flawlessly. It starts with obsessive hand hygiene. It means using contact precautions for any known resistant isolates. And because this is so often a device-related infection, it means being absolutely meticulous with your line insertion bundles and, again, getting that source control done fast. And all of this has to be supported by a really strong antimicrobial stewardship program.
But here's the final and maybe the most crucial point. We could have the best hospital infection control in the entire world, and it still wouldn't be enough on its own. Mammaliicoccus sciuri is the textbook definition of a one health problem. It's incredibly common in livestock, in our pets, in wildlife. This massive animal and environmental reservoir is constantly feeding new strains and new resistance genes into the human population. The only way to really control it is for human health, veterinary medicine, and agriculture to all work together. And that really just leaves us with this one big nagging thought. If Mammaliicoccus sciuri was hiding in plain sight this whole time, acting as this incredible reservoir and gateway for resistance, what else is out there? What other seemingly harmless environmental bugs are we completely overlooking right now? And that's our deep dive into the complicated world of Mammaliicoccus sciuri. You can go check out all the research for yourself in that linked notebook document. If you want more explainers just like this one, please make sure you subscribe to the channel. And I really want to hear from you, especially the microbiologists watching. What does your lab's protocol say when you get an oxidase-positive cons? Drop your experiences and your institutional protocols in the comments below. Thanks for watching.